You need, oh,
Sorry, what was
no,
that?
that was what I was going to ask, if it needs to be recorded. Okay, perfect.
Okay, word. All right. Hi, everybody. For any of you watching this, this is 3-1, cell signaling and receptors. So, I'm gonna kind of skim through the first part pretty quickly because I think all the signaling pathways are a little more important, but just a brief introduction to how these signals work. Cell signals, sometimes called ligands, they come in two kinds, hydrophobic and hydrophilic ones. The hydrophobic ones are the ones that could pass by the cell membrane very easily because, of course, the inside of the cell membrane is hydrophobic. So these kinds of signals can diffuse straight through the membrane. Hydrophilic ones, however, they cannot diffuse through the membrane, so they're going to need like some kind of channel to get them through. Like I'm sure you've heard of like ion channels and stuff. They're going to need like some kind of channels to get into the cell, or they're going to need some kind of receptor, like the image right here. This ligand is binding to this receptor, which allows the channel to open. Things that affect. Okay, receptors, you could have extracellular receptors, which are just receptors that stick on the outside of the cell. So kind of like here, there's a receptor on the outside of the cell, or you could have intracellular ones. So as the name implies, it's just a receptor that's inside the cell, like this one. Okay. Okay, so there's two main kinds of receptors that we're definitely going to be talking about later. So just a brief introduction to these. There's the RTK, receptor tyrosine kinase, and the way that this receptor works is these guys are just sitting around, they're just loafing on the cell membrane until a signal comes. When the signal connects to the RTK, they come together and they dimerize. So notice that they're far apart here, but when they connect to the signal, they dimerize. It's these signals coming together that allow them to autophosphorylate. So when these receptors come together, they phosphorylate each other. And now that they're phosphorylated, other proteins are now able to come and bind to this RTK receptor. And of course, we'll be going through some examples of what this looks like. Another kind of receptor that we'll be talking about is the JAK receptor. So it looks similar to RTK, but they're pretty different. It's not an RTK. The way that JAK works, JAK stands for just another kinase, which I think is hilarious. But the way that it works is, again, these guys are pretty far apart. A signal comes and causes them to dimerize. Now, in RTK, when the tyrosine receptors dimerize, they autophosphorylate each other. However, here, when the receptor comes together, it's not the receptor phosphorylating each other. It's these two proteins called JAK1 and JAK2. So again, it's not the receptor that phosphorylates anything. It's the proteins that are connected to the receptor. They cross-phosphorylate each other. So when the receptor dimerizes, JAK1 and JAK2 cross-phosphorylate each other. They activate each other. When they activate each other, they're then going to add a phosphate. They're going to phosphorylate the receptor. So again, the receptor is not doing any of the phosphorylation. It's just being phosphorylated in this case. And now that the receptor is phosphorylated, it, like the RTK pathway, it's now susceptible to having other proteins come and bind to it. So just bear these in mind, and we'll give examples of what these look like in real life. Okay, so two main outcomes for signal transduction. You could have a fast response, which is like a signal binds to a receptor, which has like some cascade event, and it changes the shape of a protein, for instance. If you change the shape of a protein, you change its function. And so that's an example of something that can happen very quickly. Another example, something that might take much slower, is a signal binds to the receptor. It activates some protein cascade, activates, I don't know, some transcription factor that goes to your DNA. It has to transcribe something. So that's obviously much slower. But these are two generally different outcomes. Oh, and Rachel, if you have anything you want to add, or yeah, just feel free to interrupt me at any time.
Okay. Yeah, no, you're good.
Okay, word. I'd ask for questions, but there's no one here. So you could pause, I guess, and just question yourself. Whatever. Okay, so signaling is obviously super important to the cell. It's how it does everything. So cells need signals for it to just stay alive. It receives signals to grow or divide, to differentiate something. If this is like a stem cell, it needs the different signals that this stem cell will get will determine what it differentiates into, so a neuron or a smooth muscle cell, whatever. If a cell stops receiving signals, it will automatically die. So it needs signals essentially just to survive. Cells always need a constant survival signal to stay alive. If they stop receiving those survival signals, or if it just stops receiving signals at all, it's going to go straight into programmed cell death. This is one of the reasons why trying to grow cells in vitro, so outside of the body, is pretty hard. Because again, that cell needs so many different kinds of signals from the environment that is very, very hard to emulate inside a test tube or on a petri dish or something. So it's very hard to grow stem cells in vitro, as opposed to when those stem cells are inside the body. Same signal, same receptors. Okay, so one signal does not signal for like one specific outcome. You could have the same signal, you could even have the exact same receptor, but depending on where this receptor is, like depending on what kind of cell it is, you'll have different outcomes. So acetylcholine is a very popular signal. Notice that acetylcholine could bind here to this receptor. This receptor is the same as this one, which is the same as this one, but they all exist on different cells. So in a heart muscle cell, the acetylcholine will cause it to contract. In whatever cell this is, the salivary gland, it'll cause salivation. On skeletal muscles, it'll cause it to contract. So one signal and one receptor is not exclusive to like one particular outcome. It could have several different outcomes depending on what the cell is. Okay, so different ways that cells talk to each other, or I guess different transduction pathways, is cell amplification. So mind you, if a ligand connects to a receptor, that receptor is going to activate like whatever cascade of proteins. But one thing that it does is a thing called signal amplification. So if you can imagine that this ligand and receptor activates only one protein here, which then only activates one other protein, which activates only one, you can imagine that this is going to take a really long time to do if it's only activating one protein at a time. So to account for this, signal amplification is the idea where it activates multiple proteins, which these three guys activate three more proteins, which each of these activate three more proteins. So it's really amplifying the signal. And that's how a signal can go through the cell very, very quickly, as opposed to just one protein at a time. So that's the essence of signal amplification. There's also crosstalk, which is one pathway is not exclusive to just one outcome. So one of the pathways that we're going to talk about is the GPCR pathway. Notice here in GPCR, we'll talk a little bit about what this is, but there's the G-alpha protein. G-alpha can activate the PLC, it could activate PI3K, it could activate RAS, it could just do like a bunch of other things relating to other pathways. So G-alpha, which is mainly found in the GPCR pathway, is not just exclusive to the GPCR pathway. G-alpha can go into the cell signaling pathway. It could go into the cell program cell death pathway. That's essentially what crosstalk is. It could go into other pathways and just do its thing there. Any questions? Just kidding. Okay.
I have a question.
What's up?
No, just kidding. Yeah, how do we want to show those videos? Do we want to?
Oh, I didn't realize they played. I was just gonna let them watch it in their own time.
Yeah.
I wasn't gonna like play these.
Yeah, that's fair.
These are just video examples if you guys want like a visual illustration of how it works. I think they're also available. I know Laura posts a lot of supplemental material, diagrams, videos to help you better understand these things if you're like visual auditory learners. So here are just a few video examples. Rachel and I have seen these like 1,000 times, so I don't think we're gonna watch them.
I highly recommend looking at videos or drawing them out, because especially with pathways, with all the different steps, drawing them out or watching a visual video of it definitely, well, at least for me, it helps kind of ingrain the order of the steps.
Yeah, same. I'm a very visual learner, so I always turn to videos when I don't like really understand something. Having that image in your brain just obviously helps you understand it, which like you can see here, if you're kind of confused about my explanation of signal amplification, you have like, again, like just this video reference to, oh geez, go back to, oh gosh, what have we done? Okay, so.
Yeah, I can. Do you want me to? I can take over.
Go for it.
Okay, so the four pathways we'll be focusing on are the RTK, RAS, MAPK pathway. The spiral pathway, which is the AKT pathway, the JAK-STAT pathways, and the GPCR pathways. So the RTK, RAS, MAPK pathways, we'll start with this one. So it's in the name. An RTK is a receptor tyrosine kinase, and that's these long blue things that Sebastian's pointing to right now. So basically, how it starts is Sebastian talked a little bit about this already, but the signal comes in. It's in the form of a dimer, and it comes in and binds to the receptor tyrosine kinase, which is originally inactive. Once it binds, it's now activated, and because the receptor tyrosine kinase has this intracellular kinase domain, so they become active once the signal binds because it causes a conformational shift. So that's the first step. Signal binds, you get a conformational shift in the RTK, and that makes it active. So once it is the kinase activity of the RTK is activated, it can now autophosphorylate, which is what Sebastian mentioned before. And that, see these yellow P's are the phosphates. So now this is a fully activated RTK. And you can go to the next slide, Sebastian, which creates these binding sites for proteins. So the next step in the RTK pathway is this adapter protein called GRAB2, which is right in this dark blue protein, binds to the phosphorylated tyrosine. And it binds to this through what is known as an SH2 domain. So it binds through the SH2 domain, and many signaling proteins have SH2 domains. And SH2 domains are basically just phosphotyrosine binding domains. And GRAB2 is also called the adapter protein, as you can see, like in the diagram, it's labeled as the adapter protein because all it does is it's a binding site for this RAS activating protein, which is also known as SOS. And SOS is also a RAS-GEF protein. So basically what this does is it binds to GRAB2 after GRAB2 binds to the RTK. And it binds to GRAB2 through an SH3 domain that is located on GRAB2. So SOS does not have a SH3 domain. It binds to GRAB2's SH3 domain. So just to recap, because I know it's a lot, GRAB2 binds to the RTK through its SH2 domain, and then SOS, or the RAS activating protein, binds to the GRAB2 through GRAB2's SH3 domain. Now, SOS has something called the PXXP motif. Basically, it uses this to bind to other proteins. So basically GRAB2 is the SH3 domain, SOS has the PXXP motif, and they bind to each other through the domain motif interaction between SH3 and the PXXP motif on SOS. So moving on to SOS's function, it's a RAS-GEF, so it activates this RAS protein. So as you can see in the diagram, when RAS is bound to GDP, it's inactive. And then so what SOS does is it's a GEF, so it swaps GDP for GTP, activating RAS. So now RAS is activated. And then just a quick little side note, if you notice, RAS is bound to the membrane through this little lipid thing. So it can only move laterally throughout the cell. It cannot move like up or down or whatever because it's bound. So so SOS activates RAS by helping it bind to GTP and let it go of GDP. So this is the rest of the pathway. So now RAS is activated, it's bound to GTP. So it can go on and activate the MAPK pathway. So it goes down in numbers, is like how I like to think of it. It goes 3, 2, 1. So you start with MAPK3, which is what RAS GTP activates. Just a quick side note though, RAS is not a kinase, and it's not known how it activates MAPK3. So it does not phosphorylate MAPK3. All we know is that it activates it. We don't know how, but we do know that it does not phosphorylate it. So basically, MAPK3 is now activated, and then it can phosphorylate MAPK2 at 2 different positions. once MAPK2 is activated, it then activates MAP kinase by phosphorylating it also. And then this activated MAP kinase can go on and phosphorylate whatever function it needs to do in the cell, whether that be a protein or a transcription regulator, a transcription factor. And you get these two results, either a change in protein activity or a change in gene expression. And that's basically it with the RAS MAPK pathway. This is just a summary, basically everything I said. So just some main ideas. You have the SH2 domain, SH3 domain, the PXXP motif. SOS is a RAS-GEF. Like if you remember from nuclear import and export, we have RAN, GEF, and GAP. It's the same type of idea here. And just to remember that RAS does not phosphorylate MAPK3. And then this is the whole pathway all together and your end results. And that's basically it for the RTK RAS MAPK pathway. Any questions? No? Okay.
Actually, do you remember if MAPK3, it doesn't phosphorylate MAPK2, it just activates it?
I think it doesn't it phosphorylate? Wait.
Does it? I'm not sure. I know this definitely doesn't phosphorylate MAPK3. I don't
Right.
remember if the MAPKs phosphorylate each other, like going downstream.
So it does, yeah. So
Oh, it
MAPK3 does
does? Okay.
phosphorylate MAPK2.
Okay. And then
yeah.
K2 phosphorylates K.
K,
MAPK.
yeah, just K.
Got it.
Also, quick note for people watching, in your answers, please don't write MAP KKK. It just doesn't sound right. Just say MAP K3, and MAP K or MAP and then MAP K2 or and then MAP kinase, whatever. Just don't use the letters for that one.
Yeah.
I think it's a little obvious. Just write MAP K3 on your exams.
You can write MAP kinase. Just kidding.
Yeah, you can write it out, but just don't
do that. No.
abbreviate it as a hate group, please. Thank you very much.
Good. Okay. Which is the pathway that you wanted to do again, Rachel? Was it
that one? The one I just did. I don't care about the other ones.
Oh, okay.
I mean,
okay.
I'll do whichever ones. I don't care. Like
yeah,
I care about
that
them, but like
beautifully detailed. Jeez, like I don't know. If
I know that's because I've had that one down.
No, so
but yeah, if you, do you have a preference? Which one do you want to do?
No, I don't mind. I
just switch off then.
Can't give the level of detail that you just gave, but
no, you're chilling. You're chilling.
Okay, word. Okay, so survival signaling. This is an example of another RTK dimerization receptor. So again, the signal comes in. The signal finds these RTKs. They dimerize, autophosphorylate each other. And now because they have a phosphate group, they're now susceptible to having other proteins come to them. So one of the proteins that approaches it is this PI3 kinase. Anything that binds to this RTK receptor, anything that binds to a receptor tyrosine must have an SH2 domain, as Rachel mentioned before. So this protein, the PI3 kinase, it is holding onto the receptor, the RTK receptor, because of its SH3 domain. So just to elaborate a little bit on the SH3. So what it does is it's now that it's active, now that it's bound to the RTK receptor, now that it's active, it's going to phosphorylate this thing. I forgot the name of it, inositol phospholipidase, but if I remember correctly, this had a name. Do you remember?
PIP3.
PIP3, yes.
PIP3.
Okay, perfect. Okay, sweet. So PI3 kinase is going to phosphorylate PIP3. PIP3 already has 2 phosphate groups on it, so it just needs a third. This guy, the PI3 kinase, is going to give PIP3 its third phosphate. Okay. Anything you want to add about this, Rachel?
No, it's basically it.
Okay. Okay, so once this guy adds the phosphate group to PIP3, there's another protein called ATK. Oh, sorry, AKT. AKT is going to find PIP3 that has 3 phosphate groups attached to it, and it's going to attach itself. So forget these guys for a minute, just focus on the AKT right here. So again, AKT is just like loafing around, it's not really doing anything until PIP3 has its 3 phosphate groups. Once it has its 3 phosphate groups, AKT is then going to find this PIP3 and is going to connect to it. But it's not functional yet until it gets phosphorylated by 2 other proteins. So protein kinase 1, protein kinase 2. I don't really know where these guys come from. They just come from somewhere. And they add a phosphate group to AKT. Does that sound about right, Rachel?
Mhm.
Okay.
Yep.
Okay, so once these two proteins find AKT and they phosphorylate it, now AKT is active. So AKT only becomes active when it gets double phosphorylated by these two kinases. Okay, so active AKT is going to find this complex. This is the bad BCl2 complex. This is bad, the blue part. This is BLC2, that red part. So as a complex, BCl2 can't do anything. It's just kind of stuck to bad. But when active ATK finds this complex, it's going to phosphorylate it, I believe. Yeah, it's going to phosphorylate bad. When it phosphorylates bad, bad then releases BCl2. And now that BCl2 is free, it's going to do its thing and it's going to inhibit programmed cell death. I think it clogs like holes in the mitochondria or something like that.
Yeah.
So again, if BCl2 is connected to bad, BCl2 can't do its thing because it's occupied. So in that case, the cell will undergo programmed cell death because BCl2 is prevented from doing its job. It gets released from bad when AKT is active and phosphorylates bad. Okay. This is a summary of pretty much everything I just talked about. And here's the whole pathway in its entirety. So just real quickly, signal comes, these guys dimerize, it autophosphorylates each other, which allows PI3 to find this receptor. Active PI3 kinase is going to turn PIP2 into PIP3. Phosphorylated PIP3 is then going to have AKT attach it to itself. Oh, okay, now I see where these guys are coming from. Okay, so PIP3 is AKT is going to find PIP3, and it's going to sit there. When AKT is phosphorylated twice by this protein and this protein, it's then going to leave PIP3. It's going to find the bad BLC2 complex. It's going to phosphorylate bad. Bad is going to release BCl2, and BCl2 is going to do its thing and prohibit apoptosis.
Bad is bad.
Yeah. Yeah,
when bad is not inactivated, it is leading to cell death.
Bad is bad. Thankfully, a lot of these names are pretty intuitive.
Yeah.
They did something right.
Yeah, seriously.
Okay.
All right. Now the JAK-STAT pathway is kind of similar to the RTK pathway, but a little bit different. So the mechanism, so a big example that Laura likes to use for the JAK-STAT signaling pathway is that this example that she shows from the textbook leads to milk protein production from breast milk for lactation. But JAK-STAT signaling is also used for cytokine signaling, cell division, and lactation, like I said. So JAK1 and 2 are in the family of tyrosine kinases, but they're non-receptor tyrosine kinases because they have the tyrosine kinase domains that are similar to RTKs, but they are not an RTK. So try not to get those confused. But so basically JAK1 and JAK2 bind tightly to the receptors. So these green things are the receptors that they are binding to. So if you see JAK1 and JAK2 are these blue like oval things, they bind to these receptors. So in this example, like I said, it's for lactation. So these are prolactin receptors. So then the prolactin signal comes in and binds to the receptors. They dimerize similar to the RTK. And then what happens is these JAKs are now activated. And what they do is they cross phosphorylate each other and then the receptor. So if you see these like little red arrows, they become activated, and then JAK1 phosphorylates JAK2 and JAK2 phosphorylates JAK1, and then they both, once they are activated and phosphorylated, can phosphorylate the receptors. So once the receptor is phosphorylated, the STAT proteins come in, which are these like weird orangey pink things. And they bind to the receptor also with an SH2 domain. So if you remember, GRAB2 has an SH2 domain as well. And then once they bind to the if you see that they bind to the first phosphate on the receptor, then once they bind to that through their SH2 domain, the JAKs then phosphorylate stat at this second location. So it comes in, binds to one phosphate, and then gets phosphorylated by the JAKs. So once they're phosphorylated, they change conformation and can dissociate from the receptor. And then what happens is they form this dimer by binding each to their own, the phosphate on the bottom. So they let go of this top phosphate and then bind, like, they see how one kind of flips, it binds to they each bind to the phosphate that was put there by the JAKs. So when they form this dimer, they can now enter the nucleus and act as a transcription factor. So they can regulate DNA transcription, which affects the transcription, in this case for lactation, of milk protein genes. But like I said, it can also affect cytokine signaling, which is an immune response, and cell division signaling. So they are now activated, and they enter the nucleus through importin, like the nuclear localization or nuclear import that we talked about. Importin brings them in that same mechanism that we talked about. And then it can bind to the DNA and affect transcription. So it's a pretty short pathway, but that's basically it. This is the summary. It's a lot of cross phosphorylation, and ultimately it leads to the transcription of certain lactate prolactin genes. Prolactin's a hormone, I'm pretty sure, but yeah. And yeah, this is just, again, the summary.
Okey-dokey. Last one. Okay, the GPCR pathway. I'm pretty sure it's involved with like a whole bunch of things. I don't know exactly what, but I know that this
It's
guy's
in
pretty involved.
a lot of things.
Yeah, this comes up like all the time. But essentially how it works is you have your G protein coupling receptor just sitting right here by the membrane. This G protein receptor is connected to G alpha, which is connected to G beta and G gamma. So right now they're just a complex. They're just sitting like this, and nothing is really going on. When a signal comes and binds to the G protein receptor, it's going to activate its GEF function. GEF is the guanine exchange factor. So what it's going to do is G alpha here, notice that it's connected to GDP, it's inactive. But again, when the signal activates the G protein receptor, it's going to activate its GEF function. It's going to scoop out the GDP and allow GTP to diffuse in. When GTP diffuses in, G alpha disassociates itself from the G protein receptor, as well as from G beta and G gamma. So again, when G alpha lets go of its GDP and allows GTP to diffuse in, it's then going to do whatever response it may be. I know this is pretty vague. Like we mentioned, this particular pathway is involved with a whole bunch of things. So this has like a bunch of different responses that it could do.
It's in almost every cell, to be honest.
Yeah, it's everywhere. Even like I was reading like random primary literature for a random class, and it was talking about this pathway. I was reading another paper for like a different class, and again, talking about this pathway. So
yeah,
involved,
if you're in bio, you're going to see this probably forever.
Yeah. So active G alpha is just G alpha that's connected to GTP. It's going to go around. It's going to do its thing. So once the cell no longer needs G alpha in its active state, another protein comes by. Where is it? Oh, the RGS. So RGS is going to, so again, this is when the cell doesn't want G alpha doing its thing anymore. It wants to stop G alpha from working. What's going to happen is RGS is going to come, it's going to find G alpha, and it's going to activate its GAP function. GAP is the GTPase activating protein. So when RGS comes and finds G alpha, it's going to activate its GAP function. It's going to hydrolyze GTP into GDP. And when that happens, again, G alpha in its inactive GDP state is going to come back to the G protein coupled receptor. It's going to bind itself there. G beta and G gamma are also going to bind to G alpha, and it's just going to sit there in its stationary state again. And of course, the cycle just repeats itself. Okay, anything you want to add to this one, Rachel?
No. That's basically it.
Word. Okay. Oh, this is another diagram.
She uses this one, or well, when I had her class, she used this on the exam, and we had to like fill it in the blanks and stuff. And she took,
Really?
yeah, it was horrible though because it
Oh my God, I
wasn't like
remember this.
colored. So it just looked like a bunch of like dark blobs because like the blue ink came out black too, so we couldn't see like what alpha, beta, and gamma was. It was, oh my God, it was a mess.
Dude, I think I remember seeing that on the final. I remember all of these subunits were all like the same color. They're all black. And
Yes.
I was like, God damn it, I
Yes.
can't see a
And we
single
had
thing
to
on it.
like fill in, like write what each one was, like, oh, the ligand nucleotide exchange factor, blah blah blah, and then
Yeah.
dissociation, blah blah blah. And, oh my God, I remember that so vividly.
I feel bad for the TAs that had to read my nonsense. I just drew
Me too.
yeah. I was like, good luck to whoever's reading this. Oh well. Okay, so pretty much done with this one.
Yeah.
If you guys have any questions about this or any confusions, of course, feel free to email Rachel or I. Our emails are here. Jesus, we email all the time, so I'm sure you can find it pretty easily. But
Yeah.
just in case, here are our emails. Feel free to reach out anytime.
Yeah.
All
Thanks
right.
for watching.
Yeah. See ya.
Woo.